Hi, everybody. Last time we talked about how you can take carbons, add functional groups to them to form subunits of larger molecules. And the specific example we did was an amino acid. And you may remember that what we started with was a carbon. And on one side of it, we bonded an amine group, which is an NH2. And on the other side of it, we bonded a carboxyl group, or a carboxylic acid group. Amine, acid, amino acid. Okay, now what I haven't drawn is all the complexities of those, but we do have on the board three amino acids here. There's one, here's another one, and here's another one. What I have not drawn are the R groups or side chains, and I'm going to leave them for the most part blank here. So I'll just throw in R, 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 and that could be any one of more than 20 different varieties up there. So why have I drawn three in a row? Well, the idea here is a protein is a polymer of amino acids, a long chain of them that has a function. And so what we need to do is take this amino acid and maybe covalently bond it to this one, and then covalently bond it to the next one in line. And you may remember this from your biology or living earth class that the big machines that do this in your cells are called ribosomes. At least they're a big part of that story. So the ribosomes are going to have an enzymatic role in covalently bonding this amino acid to the next, etc. So, but how would this look on a sort of chemical diagram? Well, the way you polymerize things in your cells typically is by doing a process called dehydration synthesis. This may sound familiar. Dehydration or dehydration synthesis is the removal of water. And so I've arranged these three amino acids in linear form. Okay? And what you can see is if I take the carboxyl end of one amino acid and put it next to the amine group of another one, you can actually see two hydrogens and an oxygen ready to go. So enzymatically, if you have a structure ready that's able to pull this off, here we go. We take the water, remove it, off goes the water. I could draw the water, I'm not going to. And we put in a new bond. There we go. And now we have two amino acids covalently bonded together. We could do it again. So again, the beginning of the amino acid is the amine group. On the far side is going to be the carboxyl group. So if we take the carboxyl of one, put it up next to the amine of the other, remove H2O, and form a new covalent bond, boom, you have now created a short polymer. So now we have three amino acids all covalently bonded to each other. This is called a peptide, or if it's longer, a polypeptide, for a polymer of amino acids. So the vocab gets kind of annoying. This is just a covalent bond. You'll see it referred to in textbooks as a peptide bond, which is just a covalent bond that is formed between amino acids. So let's not get lost in the vocabulary here. It's just a relatively stable bond. It's a covalent bond between the amine group of one amino acid and the carboxyl group of the next. So now we have a chain of amino acids called a polypeptide, and the longer it goes, the more functional shapes you can make. One thing that is worth noting about this before we get into any of those structural issues is that this is directional. And what I mean by that is there's kind of a beginning and an end, right? And that matters in terms of structure. And it matters in terms of genetics, which we'll get to later in the year. The beginning of the protein is this end. So we're going to designate this as the beginning because this is the first one that comes out of the ribosome, honestly. And so this is the amino end or the amino terminus. If you want to shorten that up, it's the N terminus. So the N end, that's going to be called the beginning. We're going to consider that the beginning because it really is. So the N term or N terminus is at one end. The other end of the molecule, if you notice, there's going to be a carboxyl group. So I can circle that up and say that's the carboxyl or C terminus. Okay, so now we have our two ends, the amino end, the N term, the other end, the carboxyl or C terminus, and we have a directional molecule. What we'll talk about in future videos and lectures is we'll talk about how you can actually take this long chain and why it's going to start actually folding up. Why are there going to be sort of bonds formed between, say, this part and something like this further down, or maybe between our groups. And that's where we get into the complex shapes. That's it for this time. See you next time.